Biology research at Microsoft is focused on three main areas: molecular programming, synthetic biology, and stem cell biology. At the intersection of biology and computing, there are implications for health, medicine, and efficient computing techniques. The field of biological computation is in its early days, and there's still lots of work to be done. Colin Gravill works in the computational science group at Microsoft Research, and on today's episode he explains how Microsoft is investigating biology with the same curiosity and pragmatism that Microsoft investigates computer science. I'd love to do more shows at the border of software and biology, and if you have suggestions, please send me an email, jeff@softwareengineeringdaily.com. I have a persisting interest in both biology and software, so I'd love to hear your thoughts on that. I hope you enjoyed today's episode. Continuous integration gives you faster, safer software delivery. With a continuous integration tool like SnapCI from Thoughtworks, the members of your team can push changes independently of each other, and they can all see their new builds running against different phases of tests before those changes make their way into production. The fastest moving companies that I've talked to on Software Engineering Daily are all using continuous integration. SnapCI from Thoughtworks is available to anyone. And if you go to snap.ci/softwareengineeringdaily, you can check it out for yourself and support Software Engineering Daily. With just a few clicks, I had my own continuous integration set up for some projects that were just sitting in my GitHub account without continuous integration. I got continuous integration up quite easily using SnapCI. If you want to be that hero at your company that starts moving your organization towards deploying often, more confidently, towards that DevOps dream, start working with SnapCI at snap.ci/softwareengineeringdaily. Your coworkers will see you working with SnapCI, and they will fall in love with it themselves. Oftentimes, it takes somebody at a bigger company to go out on their own and say, okay, I'm going to roll out CI even though nobody else at the company is using it. And maybe that hero at the company is going to be you. So check out snap.ci/softwareengineeringdaily. And thanks to Thoughtworks for being a continued sponsor of Software Engineering Daily. It really means a lot. Colin Gravill works in the computational science group at Microsoft Research. Colin, welcome to Software Engineering Daily.
Hi there.
So your research group at Microsoft is focused on three main areas: molecular programming, synthetic biology, and stem cell biology. And I'd like to get into each of these areas, but we should start more abstractly because there are so many unanswered questions in biology, and I want to get a feeling for how you approach biological questions from the standpoint of computer science, of programming. Is there a certain class of questions that are well suited today to being solved with computation? Because biology is so complex. There's so many moving pieces and so many unknown things today. What are the kinds of questions that we can answer today with computation?
Yeah, so we're focusing on these three different kind of example areas, and there's two big questions that drive us. One is trying to understand what biology is doing now, and then the other side of it is how might we be able to fabricate biological systems. So part of it's inspiration, natural scientific curiosity, and then the other side of it's engineering. So how might we be able to build systems that are useful to society, and do that using the power that biology has and the scale and efficiency.
How has the frontier of the questions that we can begin to answer and the things that we can study in biology, how has that expanded in recent years?
So one of the big changes, and this is kind of deep on the biology side, is single cell data. So what traditionally might have been done is studying populations of cells or biological systems. But what we've been able to do with like advances in instrumentation and computational methods is identify what a single cell is doing and multiple individual cells and see how that the dynamics of that cell correspond to populations. So it's quite an interesting challenge both to understand what a single cell is doing, but also how it negotiates and works with other cells to achieve a goal.
What are those technological advances that have allowed us to zoom in on a single cell?
So some of them are like microscopy methods, so also imaging methods, so we can track much better what's going on. So computer vision systems, these type of things that a lot of my colleagues in other groups, like who work on things like the Kinect and HoloLens, we can also turn some of those types of methods onto tracking what cells are doing, understanding what behaviors they have, and then feeding those into the kind of computational models we build to try and understand what they're doing and how we might be able to do similar things.
Do you think of the human body as a kind of computer?
We think it's a useful way to think about it. It's an information processing system. It has inputs and it tries to achieve certain things. What the origin of that is, it's a very philosophical question, but we find it a useful way to think about what's going on, and it's led to insights. As to its very nature, it's kind of an open question.
Are there ways that you can think of that break down that analogy? Like, I'm sure you've thought about this question a fair amount. So, how is a biological entity significantly different than the computer systems that we deal with today?
I mean, in general, you have these individual processing units, and even with like the rise of multi-core systems, you don't tend to have so many individual processes, especially not to the scale of biology, that you have these autonomous units. Whereas in computing systems, you generally have got the inputs and they collaborate, and that maybe answers a question you have about why people are using your website more or less. Whereas biology, the individual cells might be carrying out a program that they do in concert. So it's a lot more like actor models or large-scale distributed systems, but on a scale much bigger than software engineers are dealing with on a day-to-day basis.
Even those software engineers who are operating a data center and or an Azure cluster or something, you think it's still a scale that is not close to what a biological system is processing?
So on the human body contains trillions of individual cells, and each of those cells is very complicated. So if you do the kind of comparison between one of the newest Intel chips that has billions of transistors, we're several orders of scales bigger than that just on a single cell, just based on the number of cells in the body. And then there's huge amounts of complexity of how they interact. And it's not this kind of digital on-off. There's significant signals going in between about what's going on. So it's just a lot bigger and wider. But interestingly, we can use some of the similar software methods that colleagues use on distributed systems on biological systems.
Such as consensus, which we will get into.
Yep.
Is that right?
Yeah, so one of the algorithms we've published, so our kind of primary aim is publishing scientific breakthroughs. We also want to feed that into these engineering ideas, but one of them is this idea of the approximate majority calculation. So control theory has these ideas that if you have different agents, they need to come to a consensus whether something is true or false, and they might have to do that in real time. And you could take an example in biology. If there's some population of cells, and some of them seem in a bad state, so they might indicate cancer, you might want to indicate, is the overall population a large tumor or is it a small infection? And your choices how to behave would be different in those cases.
Okay, so we'll talk about more of that a little bit later, but while we're still talking at a high level, why is the area of medicine and biology and this kind of research, why is it useful to Microsoft?
So we see it as a big opportunity to just kind of explore where we can get to next. The industries that might exist later are just phenomenal. In some senses, the whole computing industry is built on programming silicon-based devices. We see situations in industry where if you can have intelligent control over chemical fabrication and you can efficiently design bacteria that can create those products, that's an industry that's even bigger than programming silicon chips. The potential is enormous, but the question is, how is it possible? How can you do it efficiently and effectively?
And it sounds like the barriers keeping us from applying some of these computational concepts to biology, at this point, those barriers are more rooted in science than they are in policy. We're not at the point where it's kind of like with drones, where with drones, like the technology is really getting there, and we could imagine, very reasonably imagine, production systems with drones flying around, if not for the policy. But with biology, we're not really there yet, right? It's not like we have policy that's significantly impinging on our ability to progress. Is that right?
I mean, there are definitely some of the stem cell type work that we collaborate on that obviously has kind of questions and implications. That there are treatments that are happening now based on stem cell therapies, and the challenge is we don't really understand why they work and what the times when they don't work. And some of our work is helping contribute a fundamental understanding to that. So what we hope is that we're helping make it more feasible to give better treatments. So that's probably one area where there's kind of policy and direction of, in some senses, we're trying to re-engineer human bodies. We're doing it in a very indirect fashion. Some of the other areas where we do more fundamental long-term research are around computing with molecules. That's a lot more speculative. And I'd agree there that we have some technology questions to answer first.
Okay. So what is at the intersection of machine learning and biology?
I mean, so part of it is there's this just enormous corpus of data that we can acquire, but then feeding it through our models, and so efficiently choosing the models to do that. So, I mean, one of the key techniques we use is it's an idea called MCNC, Monte Carlo Markov chains. So, and you can think of this as taking a spot in the space of possible behaviors in biology and running a computer simulation from there and seeing whether that simulation gets you to a good result. And if you do this millions and millions of times, you can optimize your model to be more like reality. And so this is kind of one of these machine learning techniques for adaptively understanding. I mean, it's also very useful to have so many machine learning experts to help apply this process to this really interesting application area.
And what about specifically deep learning? Like, I've done a number of shows recently where we've touched on deep learning, and it seems like deep learning is really useful in these areas where you have a complex classification problem, where it's hard to define the features that would create or compose a classifier, but we have so many examples that fall under that classification that we can use deep learning to learn the features. The deep learning system can learn those features itself. For example, if you have a billion pictures of tumors, maybe you don't want to create a manual set of features that will classify a tumor, but you can just feed in all those images into a deep learning system. Are you seeing any interesting examples of deep learning?
We're definitely seeing it. Primarily in my group, we tend to work on what we term mechanistic models, so understanding how proteins and cells interact, and we tend to calibrate models on that. Where I kind of see a lot of opportunities and places where deep learning techniques are being used are the kind of imaging of input data. So if you're imaging how cells are behaving, how well perhaps they're following the programs we might have tried building and putting in a petri dish, there, the deep learning techniques make it a lot cheaper and easier to extract the signal from the system. But it's kind of on the edge of the inputs to our systems.
Can you talk about that in more detail? That sounds pretty interesting.
So a lot of this, I think there's a couple of papers that have been recently released of doing GPU-based image analysis of bacterial colonies. And so historically, a lot of this stuff is done in a very painful way. So this is one of the challenges with science, that if you can get it done with a PhD student who manually labels data, you can actually get quite far along with that process. And it's useful to do it sometimes by hand, but we're kind of crossing the scales now that we need a lot better constrained models and a lot more accurate. And so it's not feasible to deploy the number of people you need to annotate data. And by using some of these deep learning techniques and other machine learning methods, and this is an area that I'm personally an expert in, we're able to get a lot better signal for a lot less effort.
So you've got a bacon delivery service, and you need to notify your customers when their bacon has arrived at their doorstep. Twilio helps you make sure your customers get the bacon while it's hot. Twilio's programmable API lets you build SMS or voice alerts easily in the programming language of your choice, all in under 5 minutes with only a few lines of code. Now your customers get a text or a call the instant their bacon is ready. If your customers want to see the bacon frying on a hot pan, Twilio has video APIs and SDKs for the platforms that you know and love. Learn more at go.twilio.com/podcast and get an additional $10 when you sign up and upgrade your account. That's go.twilio.com/podcast. You will only pay for what you use, and it costs less than a penny to send a text. Get started at go.twilio.com/podcast. Get your bacon delivery service cooking with Twilio's APIs for voice, SMS, and video. Okay, I want to start to talk about some of the areas that your team has looked into specifically. The first one is molecular programming. What is molecular programming?
So this is the kind of building up version. So we sometimes term what we do as nanotechnology. So rather than building smaller and smaller robots, instead what we're doing is taking a double-stranded DNA helix and cutting it in the right places. And so this is a series of chemical bonds on the nanoscale. And we've cut them in just the right places that we can form these useful little units. And one of the things we can do, just for the sake of understanding, is we can cut them so that we can produce the equivalent AND gate. So that if we had a little fragment that represented the AND section, you'd need both of the other chemicals to be present in a solution for it to be triggered. And so what we might be able to do is bind on the output of this process to fluoresce. So this means we could plate this up on a petri dish and then image it, and we've got this equivalent to the foundation of electronics.
So there is also this term molecular computing. Can you differentiate molecular programming from molecular computing?
There might be a bit of a distinction in the sense of the computation that's occurring, and we build programming languages to do this. So we've built domain-specific languages that are kind of molecular programming in the sense of you're designing, simulating, and then the computation that comes out. I mean, these terms are a bit interchangeably used as well.
Oh, okay. Okay, so do molecules, do they act deterministically enough to build computers out of them, or do you have to build higher level abstractions out of these molecules to factor out the non-determinism? Like with transistors, we have a notion that it's a 0 or a 1, it's on or it's off, but in reality, there's a range of charges, or I can't remember the voltage or something, where it's in at the lowest level, it's non-deterministic. It's not highly granular if the transistor is on or off, but we define a range of states that indicate that it's on and a range of states that indicate that it's off. Is it the same thing with molecules, where you have a sense of non-determinism at the lowest level and you build abstractions at a higher level that factor out that non-determinism?
So this is actually a really interesting ongoing debate. So it's exactly as you say. In electronics, we have this illusion that things are binary and consistent. And it's really interesting that like ongoing work of Intel and other people are to try and get these voltages down so things are more efficient. And the problem with that is you might get higher error rates. They need to balance that. And there's an idea that we might do a similar thing in biology, that although the individual gates and reactions are noisy because all sorts of things are happening, other things get into solution and break the gates. There might be manufacturing issues. We might be able to build a level of gates and digital logic on top, despite the system being noisy. Now, the challenge with this is that it uses quite a lot of structure, and especially when you attempt to do this type of thing in cells rather than in molecules, you're putting an energy burden on the system again. And in the case of cells, that can be quite bad because cells like to evolve out burden. And so there's this alternative approach. It's a bit like analog computing, that rather than building a layer of digital logic with gates. Instead, we can embed the algorithms more directly. And so an idea of this is a bit like we were saying earlier about the approximate majority computation. So we're not building it out of individual gates as such. We've got this distributed consensus system, and the algorithm has been more directly implemented. And some of the tools are actually, this is almost their purpose. We build progressively higher abstractions, and then just like on the computer, where you compile down to assembly, we compile down to DNA as letters.
Are you saying that the purpose of, or one of the purposes of the consensus protocol that you built at the molecular level was to do this kind of, build this kind of determinism out of a non-deterministic system?
Exactly. So rather than trying to suppress noise, and there are lots of different ways to go this, and they are interconnected, but it might be possible to build some of these algorithms that more directly take advantage of biological type systems. That biology is really interesting. It has all this redundancy because in some ways that's the more efficient way, or it might be it just happened to evolve into that position and then that happened to be successful. But there is a kind of general trend towards things that exist in biology are there because they're more efficient than more complex alternatives.
So you have this system of molecular computing, which is where you use molecules to create what are effectively programmable computers at the molecular level. And then you have molecular programming, which can be used to simulate molecular systems with code. So this would be in a computer. You could simulate how molecules might work. So perhaps you can run simulations a lot more aggressively on the computer than you would be able to with with a wet lab. How do you use these two systems together, and what does the workflow pipeline look like when you're trying to run some large experiment or develop some kind of system with these two kinds of systems in conjunction?
Yeah, so what we would do, this is more the ideal workflow, then I'll talk about what happens in practice as well. Okay. That we would type in a program in one of our design tools, and it looks like a programming language with some very obtuse syntax, even worse than like APL and the like. But what it lets you do is then run a simulation of what you expect to happen. And this is built on energy models and understanding. But it's not doing anything as detailed as like atomistic molecular because that's too expensive, and even with a supercomputer, it can only run for a few milliseconds with a lot of compute. So we've got these slightly higher levels of abstractions that over time we've demonstrated capture what reality happens. We've got this prediction, and this would be a useful piece of functionality. And so we take the output of our tool that says the DNA, we might send it to a company on the internet over the web, i.e. the specification. They will, in general, try and fabricate that for us, but depending on the sequences, they might have to talk to us about it. They'll then post the primers through the postal service. They'll arrive here in the lab. We'll then take it and then build up more copies of those, and then carry out an equivalent experiment to the software experiment, and then see how well it compares.
I want to talk more about this consensus algorithm or system. I'm not exactly sure what it is. So we see consensus in computer science when you have a big distributed system and you need, for example, a distributed database. You need each of those database replicas to have an identical sense of the world. Otherwise, when people make requests to this database, to this distributed database, they might get different answers. So you need these different databases to have consensus, and the algorithms that allow those databases to have consensus are quite complicated. There's some of the hardest algorithms I've dealt with in computer science. So how do you build that into molecular machinery? What was the process, and what was the motivation for that project?
So a lot of this is, again, this idea of fundamentals, that if you can demonstrate you can do these key building blocks, you're able to notionally build anything else. We've demonstrated like several of our languages that we can fabricate a Turing complete. So in theory, you can encode any compute out of that. Now, there are questions for efficiency, as with what's the most comfortable language to work in, what's the most effective one to build. Now, it's actually kind of interesting in the case of the consensus algorithm that we published, that it's not actually a true majority, it's an approximate majority. And what this means is there's a probability of error. And so these can actually be useful in computing systems. So you have all these ideas about things like eventual consistency, and you might have somebody who connects to your website, and it might be sufficient for you to get an approximately correct answer. So the times when this might be important are if you need better performance, or you need better availability, but you need to answer the question as data flows in. Then being able to, for example, have what's more of one or more of less, and if there's only a minuscule probability of an incorrect answer, that might be tolerable depending on your system design.
What are the other things that Microsoft is doing around this molecular programming and molecular computing?
So there's a quite interesting project by a related group of ours in the US. So they're working on, rather than molecular computing, they're working on molecular storage. So using DNA as a long-term mechanism for storing data. As you can probably imagine, that they're associated with the Azure team as well. And the long-term goal for this is just the enormous potential. If you look at DNA and you imagine being able to pack enough of it in, its information density is far above any kind of conventional storage that we have. So by some measures, you can fit exabytes of data into cubic millimeters. Now you've got a question about how you get it all together and how you untangle it, but these are questions that they're looking into understanding.
Oh, that would be so much better than tape drives.
It's got potential. It'd be a lot easier to carry around all your data in some ways, but you do have to get it off as well, and so that's something that's being actively looked into. One of the interesting things actually is that team has the current published record of biggest amount of information stored, and so it's 200 megabytes, which is, it sounds very small, but these processes go up rapidly when you can figure out tools for doing it.
They sure do. So is your sense that the ways that we will be using biology as computation, do you think they'll be similar to how our bodies use biology for computation, or are the processes that we have evolved so complex and so difficult to actually untangle that we can't actually get to the root of how our body... I mean, when I think about it, it's like, you know, we have these deep learning systems that we're building these days, like image recognition systems, and the researchers who are building these image recognition systems, they will admit up front, we don't know how this is working. I mean, at some level, we understand how it's working, but, you know, we openly admit these deep learning systems, they evolve in such a way that we don't really understand the features that they're looking for. They've got so many layers, blah, 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 blah. And that's how humans evolved. That's how our bodies worked. And so it seems like almost intractable to decode how humans function from a biological standpoint. Now, what we can do is look at the primitives of the human body and just observe that there are really efficient data storage techniques and systems processing techniques, and we can say, okay, let's look at that. Let's look at the human body as proof that we can build these kinds of things. But decoding how the human body has actually evolved those systems may be intractable. I mean, do you kind of see what I'm saying? Like, there seems like these two separate paths where you can look at biology as a template and a wide open space where you can derive your own systems, and then you can look at it as something where you're really trying to replicate the way that things have evolved over time. And the latter, to me, seems far less tractable.
Yeah, I mean, so it's a really interesting question. It kind of gets to the heart of some of what we're trying to do as a group. So there are these two different takes on biology. One is the kind of bioinformatics route, where you've got observed behavior, the phenotype of an organism, and you have the genetic code, and you try and draw correlations and causation between those. We're actually, it's like you were saying, that we're focusing on some of the fundamental primitives. So we're trying to attack that problem of how this individual bits and piece of a biological system works. Like, for example, how an immune system cell assesses whether another cell has been infected or whether it's healthy. And there are a lot of imaging techniques that you can look at the fragments of proteins that are coming off and trying to build models of how the immune system works from a mathematical perspective. And we've had some really good progress in understanding estimates of that and then using that to build predictive models of how the body will behave. So we are seeing on a functional basis fragments of how biological systems work, success in building software models that are mechanistic, i.e. they're not just deep learning models, which, as you say, a lot of the practitioners in the field will say it's predictive, but we don't understand the inside reasoning. We try, where possible, to work on a basis of understand the mechanism and be able to draw connections that we could explain to a biologist and things they would recognize, because we feel it's long-term more supportable.
Sure, absolutely. I mean, yeah, like I think about the medical industry, and I know a number of people who are on these pharmaceutical drugs, or they're either it's chronically they're on the drug, like they're planning to be on it for the rest of their life, or it's some kind of short-term thing. And I always found the claims that a lot of pharmaceutical drugs to be making to be so suspect, because they tend to be claims that are just like, okay, yeah, we apply this drug and then we get this kind of outcome, and then they sort of sidestep the question of, okay, this is a causal connection, or or we have a very good understanding of this causal connection, when in fact it's more of like a correlative thing, like we've thrown enough stuff at the wall and this kind of works better than other things, like, and there's a disconnect between the consumer and the creator of that pharmaceutical practice. And this is maybe getting a little off topic, but I just bring it up to say that I agree with your perspective that it would be more sustainable to understand what is actually going on, rather than having this kind of random walk and eventually stumbling upon some sort of success.
Yeah, I mean, so we do use those techniques as well because we're pragmatic. We'll use what works. And so one good example of that is that we've built circuits that fulfill a purpose, and we've generated other circuits that are more complex versions of that, like adding in various different features that correspond to biological systems. And then you've got a question like, why is the biology like that? And we have a really interesting paper that we've used these kind of numerical methods to simulate a noisy system. So the things you find in a body, like a lot of chemicals going on and changing how the system is being signaled, and simulated that in software. And we show that the more complex versions are better and better at handling noise. So a simple circuit works in a petri dish on its own, but by adding in these specific areas of complexity, we can show that in noisy systems. Now, it's a bit of a stretch to say that we understand the mechanisms of exactly why that's happening in the biology or why that was chosen. But it is interesting that the complexity can be progressively justified at times.
An area that we haven't discussed yet that Microsoft is concerned with is synthetic biology. What is synthetic biology?
There's a lot of different definitions of this. Kind of one of the origin, the idea would be you'd be able to build up from fundamental parts, kind of equivalent biology at a larger scale. So, but other people use the term synthetic biology to be a more generic engineering of biology. The idea that you might be able to alter cells to produce stuff that you want. So this is kind of used for industrial processes. So if you want to produce a certain drug, like we've mentioned the pharmaceutical companies, most of the companies are now switching over to using biological routes. They tended to use chemical routes before, but biological routes are, in a sense, chemistry with a bit more intelligence and adaptability, that they can more efficiently and at higher yields, if you can design the biology, produce what you need.
What do you mean? Can you talk more about how those pharmaceutical research techniques have changed over time?
Yeah, so you often in pharmaceuticals trying to produce a certain compound that you've shown has like an on-target effect for some kind of disease or issue a person has. And so you know the chemical you want to produce, and there might be routes to produce that through chemistry, but they might be incredibly low yield and therefore expensive. You might need vast amounts of chemicals to produce fractions of a percentage of the target molecule that you want. And it's also kind of interesting that biology has all these tricks that it can catalyze reactions that are infeasible otherwise. And so you might know the molecule you want, but you can't easily produce it. But some other form of a biological entity like cells might happen to do a part of that pathway. And so it gives you an extra bit of a toolbox.
Can you talk about the projects at Microsoft around synthetic biology?
Yeah, so one of the things we're doing at the moment, again, it comes back to fundamentals, is Alan Turing had this really interesting paper in the 50s where he talks about the chemical basis of morphogenesis. The important point about this is that one of the things he observed is a lot of high-level macroscale biological systems, like stripes or spots on animals, can actually be explained by really simple chemical interactions. So you can imagine being able to do this if you said, like, send the dark chemicals to this part, and if you had a master plan. But what he was observing is that these systems can actually self-organize and produce this really interesting complexity. And they're all questions about symmetry breaking, about how this actually goes on in practice. But what we're trying to do is take that mathematical model and build it into cells so that we can form patterns in bacteria.
I want to talk about stem cells some. What is a stem cell, and how does stem cell biology play into Microsoft's research?
Okay, so a stem cell is a cell that can become many other types of cells and also self-renew. So the origin of your body is made up by a series of stem cells that have differentiated along a tree of different cells. And so there are parts of your body where, in some kind of bloods, that you might still have like certain quantities of stem cells. The hope with this is it's kind of the basis of this idea of regenerative medicine. If you can convince some of your cells to go back up the differentiation and become other cells, it could fix all sorts of problems. So the possibilities of this are huge. That therapies are used for people who are blind and people who have had stem cells harvested from them, or fully differentiated cells. They've been treated with chemicals, turned back into the stem cells, and had them re-injected, and they've been able to see. I mean, so this is the kind of, like, this is what science should be in terms of medicine. The problems we see is that we don't know why that works at times.
Indeed Prime flips the typical model of job search and makes it easy to apply to multiple jobs and get multiple offers. Indeed Prime simplifies your job search and helps you land that ideal software engineering position from companies like Facebook or Uber or Dropbox. Candidates get immediate exposure to top companies with just one simple application to Indeed Prime. And the companies on Prime's exclusive platform message the candidates with salary and equity up front. Indeed Prime is 100% free for candidates. There are no strings attached. Sign up now and help support Software Engineering Daily by going to Indeed.com/SEDaily. That's Indeed.com/SEDaily if you're looking for a job and want a simpler job search experience. You can also put money in your pocket by referring your friends and colleagues. Refer a software engineer to the platform and get $200 when they get contacted by a company, and $2,000 when they accept a job through Prime. You can learn more about this at Indeed.com/prime/referral. That's Indeed.com/prime/referral for the Indeed referral program. Thanks to Indeed for being a continued sponsor of Software Engineering Daily. If I ever leave the podcasting world and need to find a job once again, Indeed Prime will be my first stop. So we don't know why it works, but we were compelled to try it. What's at the root of that scientific method? I mean, because I think that's kind of gets to the heart of what we're talking about. Also, like, here is something that we don't totally understand, but we were compelled to try it. How were we compelled in that direction?
Just because there's so much motivation of understanding something like developmental biology. So understanding how you go from a fetus to a full baby and you're born, and as your body progresses over time. I mean, that type of thing is studied in simpler mammals and also simpler organisms as well. And we want to understand how that process works. And one of the key elements of that is, how does a cell decide it should differentiate into one cell type? How does it decide this is the right location? And this is thought to be like series of chemical signals and internal state of tracking what's happened to the cell in the past. And we want to know what that process is. And you can do that potentially by breaking the cells in particular ways and seeing if it still fulfills its role, or adding additional chemicals at different time periods.
Are there specific research experiments around stem cells that you're doing at Microsoft?
So we don't tend to do any of the stem cell experiments in-house. I should actually mention, although we have a wet lab here, we also collaborate with the Plant Sciences group in Cambridge, the University of Washington in the US. On the stem cell work, all the work is done externally from our lab. So there's a Stem Cell Institute at the University of Cambridge that do amazing work, and we collaborate with them on this. So what we're tending to do is the modeling and helping advise the experimentalists which experiments are the most likely to have interesting results. So one of the experiments that we've published a paper in Science about is taking experimental data about which chemical signals were needed to make the genes switch on in the right sequence for the stem cells to self-renew their population. And so the way we do this is by taking models of the genetic network and experimental observations and running it through what's termed a constraint solver or an SMT solver. So this is a piece of research that Microsoft uses for analyzing like device drivers and the like. We've turned it and applied it to biology.
Can you talk more about how did you have the insight to apply that type of tool to something so closely related to biology?
I can only explain that by my extremely brilliant colleagues.
Okay. That's very interesting. Can you talk more about how you see private institutions like Microsoft interacting with the research universities? Because it sounds like the relationship that Microsoft has with these universities, like in Cambridge or University of Washington, it sounds like it really is a relationship that caters to the strengths of both parties.
Yeah, absolutely. I mean, so we historically we focused entirely on the modeling. It's only been in the last couple of years that we've also started doing some of the experimental validation as well, and we still maintain our links with other labs on a lot of the larger scale experimental work. We just wanted to narrow the feedback loop on some of our internal designs as well. But you should definitely think of Microsoft Research in general, but us especially, as being like an academic organization. We see the long-term industrial potential with a lot of ideas, but it's kind of amazing that Microsoft has this long-term vision of ideas and giving them time to come to fruition.
Absolutely. And given that long-term thinking and the academic sensibility that you seem to have from the Microsoft research standpoint, what's your sense for how how things progress in Microsoft research versus in universities? Because I mean, in universities and perhaps government institutions, there are places where there's red tape or funding issues. Are the lack of those things desirable qualities that you kind of seek out, given that they're not as existent in an industrial setting?
I mean, it's certainly amazing to have the resources when we can demonstrate the value of it. So there have been people who have had ideas internally that they can commercialize them, and then they can get enormous amounts of resources internally to then back them up. But it's great to also be able to carry out the more speculative ideas that are going to be huge game changers in the future. So there's the work on things like the Connect that were many years in the making and now major pieces of deployed commercial technology.
Has Microsoft had any fruitful partnerships with other industrial organizations like pharmaceutical companies?
We certainly have kind of ongoing work. A lot of the ideas that we work on are these fundamental improvements, and so a lot of pharmaceutical companies come to talk to us about these ideas and how they might apply them to design of biological systems. I mean, it'll be interesting to see what happens in the future, like whether the kind of active therapeutics based on these methods are something that shows enough benefit that could be deployed commercially as well.
There are certain things that we do today in medicine that I think eventually will look like they are really like out of the Stone Age. Like they're going to look like the modern equivalent of bloodletting. And specifically, I'm thinking of the lack of personalized medicine, like the way that we treat people in terms of medicine, and it's not personalized. I think of this as an example where it's really unfortunate that we can kind of see the road in terms of personalized medicine, how useful it's going to be, but we're not there yet because we don't have the right tooling, we don't have the right, experimental techniques, or maybe you could talk more about that. Like, what is the road to personalized medicine?
I mean, so there's actually a really interesting version of that I saw, and this is very speculative, and it's not work done directly by our lab, but people took samples of a person. They applied the right chemicals to induce stem cell-like behavior, and they were able to grow what's termed a liver organoid. And so the idea of this is it's not a full organ, it's a set of cells or set of tissue that has some of the same characteristics. And one of the ideas comes exactly as you're saying to kind of personalize medicine, that if you could grow this on a person basis, you could then give the chemicals you're proposing to give to that person to this organoid instead first, and then see if there's any rejection or see what effects it has. And so the potential for like long-term drug safety and experimental technique is enormous, that rather than having to treat on animal models, you can instead treat more directly on something that corresponds to human. Because, I mean, we see the same things of press releases of cancer treatments in mice, which is great. It's good for treating cancer in mice. But a lot of those don't necessarily translate because of the enormous complexity and even variability between mice and human immune systems.
Not to mention the ethical concerns that we kind of put to the side with animal testing. I mean, testing on an organoid, there's no nervous system that it's connected to. So as far as we understand pain and how animals process pain, there's not really any of that associated with testing on an organoid.
Exactly. It would be amazing if we could move all the testing into software, but we probably need some biological models. And so it would be an amazing intermediate step of growing the models of biological systems that we want with sufficient accuracy that drugs and experiments could be tested on those as an intermediate step.
And drug discovery, I think you mentioned that the strategies for drug discovery are going from a purely chemistry basis to more of a biological basis. How do you see drug discovery changing in the near future?
So just on the shorter term basis, it's the fabrication of those drugs. So drugs that we've established are already efficient. Some of them are actually quite hard to grow. So there's a great case of this in synthetic biology where one of the precursors to an anti-malarial drug can only be extracted from a certain plant. This is artemisinin acid. And a company called Amaris had an enormous many-year project costing many millions of dollars in order to design a biological pathway that produced this compound. This is too expensive. Like most companies aren't being able to afford that. It's not cost-effective to do that. And so just from a pharmaceutical drug manufacturing perspective, then there's a lot of benefit that these synthetic biology fabrication techniques have. So longer term, there's these questions about could we design better drugs? But there's a lot more speculative questions around that.
What about CRISPR? I hear a lot about this technology. What is CRISPR useful for? Are you doing any research around CRISPR?
So this is an efficient editing technique. So you can actually just see it in some ways as an engineering technique, that it becomes cheaper to edit genomes where you want to edit them. So the origin of this is it's a kind of bacterial immune system that it uses to defend itself against viruses. It's kind of weird to think about this, that bacteria themselves get infections as well. And so this is actually a quite recent discovery, and it's become a huge thing in the engineering of biological systems in the last few years. But you can also just see it as a cost reduction, that there were techniques for fabricating the genomes you wanted, but they were laborious, and CRISPR is a lot more targeted. So this allows us to write the changes to biological systems that we want to in a cheaper fashion, and also a lot fewer off-target effects.
The current way that we do biological testing and wet labs and stuff, it reminds me of like when you see those. I mean, when I'm just like projecting myself into the future, it reminds me of those pictures that you see of people working in these huge rooms with IBM computers or with vacuum tubes and stuff. And it seems like we are going to eventually get to a point where we have these automated labs where it's like an Amazon Web Services or a Microsoft Azure for running biological experiments, and where you get to the point where you can have some kid who's in high school who can spin up an experiment from his desktop computer. Do you think is that a fanciful notion, or do you think that is something that will be a reality?
Yeah, there's a couple of companies that do a version of this. So Emerald,
right? There's an Emerald something.
Emerald Cloud Labs, I think it is. There's also a company called Transcriptic. There's also a related application called I think it's Synthetic Biology Foundry at Imperial College in London. So the UK is investing substantially in synthetic biology in general, and Microsoft Research is part of a consortium, and one of those aspects is this idea of a foundry where you can send instructions for an experiment, and those instructions will be carried out, and you'll then get the results back. So that in some ways is even better than what we tend to do at the moment, which is we send off the specification for our inputs, and then we do a lot of the experiments ourselves. It is really early days. So some of the companies involved in this, what actually happens is you get a list of instructions, and then somebody in a lab wanders around and carries out those, which I don't know if the origin of the term, like Mechanical Turk. I kind of think of it a little bit like Mechanical Turk version of biology. It helps you're not having to do it yourself, but there's still a human doing it.
Human in the loop. I mean, but that's oftentimes the place where complex automation starts, because once you have the Mechanical Turk side of things, then you've at least bounded the start and end of the process that is necessary to automate, and then the person who is in the middle proceeding with that automation can better understand, okay, what are the steps that make up this wet lab experiment that the person has commissioned me to do, and then one by one those individual steps can be automated, and before you know it, you've got a robot system doing it.
Yeah, so exactly. I mean, we have a number of robots in our own wet lab that we're trying to connect from our programming languages. So it goes beyond just the software simulation, but we want to be able to design experiments that have the highest information content. And ideally, we'd like a lot of these things to be carried out automatically. But our experimental biologists are significantly more flexible. So you have this kind of experimental phase, and then you might turn it into a standardized environment. So a bit like in computing, like you experiment with a machine how to specify what you want it to be, and then you have a standardized container and you can deploy 1,000 of them.
What are the other tools that Microsoft Research is developing and using, stuff that we have not discussed yet? Can you talk about some of the tooling that your team is working on?
I mean, in general, we tend to make all our tools available. So the areas we've talked about, there are corresponding software tools, and we tend to think it's like just good science, that when we publish, we also try and make tools available that people can run over the web. I mean, but the thing is, most of these tools are for the relatively small scale. And so one of the big questions we have is, how do we make it possible for people to use whole clusters the same way we do internally? And what we experiment with is taking exactly the same methods of analysis, but be able to run them through the kind of machine learning algorithms. And we'd like to make these things available, because it's in-house, so people could spin up all the machines necessary to do it on Azure, get back their answers, and then get on with their science.
Okay, I want to close off with a somewhat naive question, but maybe you will have some interesting responses. I find that naive questions sometimes spur complex and interesting responses, but do you think we will solve cancer? Because cancer to me seems like an example of this thing where it's really hard to tell, do all malignant cancers have some kind of quality that we can define and then deploy a drug to snipe all of those types of cancers? Or is it this thing that is so deeply wedded to our biology that we can't identify the generality of cancer, and we are reduced to this problem of developing specialized treatments for all specific kinds of cancers?
So most of the biologists in like cancer therapies tend to say that it's not really thinking of it in the right way to cure cancer, because cancer is in a sense your cells having errors and going off on rogue. It's not. You shouldn't think of it exactly as this infection that's come into your body that you want to kill each individual one. Now, that said, one way to cure the cancer is if you can selectively kill those cells that have these bugs, these errors going on, you can manage the whole process. And so one of the things that we've worked around is like chemical sensing. And so if you can do things like accurately detect a cell is cancerous, and if you take another technique, which we haven't mentioned, which is termed like DNA origami, and so this is weaving or stapling together sheets of DNA and putting therapeutics inside there, you can actually more selectively deliver those therapeutics. Now, they've shown this can work in experimental lab conditions, but this is not something that's done in humans or at all yet. But the idea would be, if you can selectively target where things are and you can recognize it accurately, and you're operating in a molecular scale, you can more accurately interact with it. Rather than, like you were saying, the very primitive technique of essentially chemotherapy drug is selective poison in your body. And because cancer cells tend to grow more rapidly, they consume more poison. And that nobody would think that's a good idea, but actually it's kind of effective, obviously.
Okay, Colin. Well, thanks for coming on the show. It's been an inspiring conversation with you, and exciting to hear about where Microsoft is going in terms of research into biology.
Great. Thanks for inviting me on.
If your organization is working on a microservices architecture, check out the microservices.com Practitioner Summit in San Francisco on January 31st, sponsored by datawire.io. The summit is a full day of talks from microservice practitioners from Uber, Yelp, New Relic, Exum, and Red Hat. Check out microservices.com/SE-daily for more information. That's microservices.com/SE-daily. I hope you enjoy the Microservices Summit.